Hey guys, welcome back to my YouTube channel MedEdophilia, and in today's video we are going to start fresh, a new chapter from botany. This chapter will help you understand all the basics from botany. So the chapter is Cell, the unit of life. So what is a cell? We all know from our earlier classes. Cell is a structural and functional unit of life. By life we mean all living things around us. Now, what is cytology? Cytology, whenever a name ends with OGY, it means study. So, cyto means cell, so it is study of cells. So, unicellular organism, that is, a single cell, is capable of independent existence and perform essential functions of life like digestion, respiration, and excretion. So anything less than a complete structure of a single cell does not ensure independent living, which means that anything less than a cell, a single cell, it cannot be able to live independently. It cannot be able to exist independently in nature. So the minimum requirement for life, for existence, is a single cell, a one cell. So let's see the discoveries made in this context one by one. The first scientist is Anton von Leeuwenhoek, who first saw and described a living cell. Okay, so there's one more person whose name is Robert Hooke, and he is connected with finding out dead cells first from the cork, which resemble like the honeycomb. So Anton von Leeuwenhoek for living cell and Robert Hooke for dead cells. So remember this difference. Next we have Robert Brown, who discovered the nucleus. So Robert Hooke, Robert Brown, don't get confused. Robert Brown is nucleus. So nucleus, we know it has the nickname control room of a cell. So with the invention of electron microscope, we were able to observe the structural details of the cell. So it is with the invention of electron microscope in which we were able to make our studies on the cell easier. Okay. Then we have the scientist Schleiden, who is a German botanist, and in 1838, so there are 4 points here. Schleiden, first the name, then he is a German and he is a botanist. And in which year? In 1838. 1838. He discovered that all plants are composed of cells. Next we have Schwann. He is a British zoologist and in 1839. Same four data here also. Schwann, the scientist name, and he is the British. He is a zoologist. And when did he find out? He found it out on 1839, the very next year. So we have four things to remember here. So what did he find out? He said that animal cells had a thin outer layer called the plasma membrane or the cell membrane. And for plants, he said, cell wall. The presence of a cell wall is a unique character of plant cells. So although he is a zoologist, he has said something about the plant cells also. So they will ask you in the question, the presence of cell wall is a unique character of plant cells. This was found out by, and the first option will be Schleiden. And we all will think, yes, he is a botanist, so this is something about plants, so he must have found it out. No. So this is clearly given in your NCERT, and that's why your NCERT plays an important role. So although he is a zoologist, Schwann is a zoologist, he had found out that presence of cell wall is a unique character of plant cells. Next point he proposed that the bodies of animals and plants both, so he is including both animals and plants, are composed of cells and products of cells. So this is a correct statement. They will give you this statement, and if they'll ask you this hypothesis was produced by Schwann, yes, this is correct. So thinking that there is plants also involved, please do not mark it as a false statement. So be careful. Okay. So Schleiden and Schwann together formulated the cell theory. But this cell theory formulated by them did not explain the formation of new cells. It was Rudolf Virchow who proposed this theory, which is most popularly called as omnis cellula e cellula. According to this, it means that old cells divided and new cells are formed from pre-existing cells. So here he is explaining the formation of new cells. He is telling new cells are formed from pre-existing cells. Already existing cells give rise to new cells. And in this way he modified the cell theory which was earlier formulated by Schleiden and Schwann, and he gave the final shape to the cell theory. So now when we say cell theory, it has two postulates. First one, all organisms or living organisms are composed of cells and products of cells. So living organism, there are plants, animals and other living organisms present. This statement was actually a statement from a hypothesis produced by Schwann. He said plants and animals. Who said this? It was proposed by Schwann. And the second point of cell theory is that all cells arise from pre-existing cells. And this hypothesis was given by Virchow. And only about plants, it was given by Schleiden. And what did Schwann say? He told plants and animals are composed of cells and products of cells, and he also talked about plasma membrane, and he also talked about cell wall, the coverings. So remember what each scientist had proposed. Okay. Now we move on to the component of cell, which is the cytoplasm. Cytoplasm is a component of cell which is present in both eukaryotic and prokaryotic cells. Cytoplasm is a semi-fluid matrix. Semi-fluid matrix, which means it's a gel-like matrix, and it occupies the volume of the cell. It fills the space of the cell and is the main area of cellular activities. It is the place where cellular activities occur. Most of the cellular activities occur, and this is an important phrase used in your question papers. And we said main arena of cellular activities. This means that various chemical reactions occur in the cell in order to maintain the cell in the living state. Only if reactions occur, we say that the cell is in the living state. Okay. We said that cytoplasm is present in both eukaryotic and prokaryotic cells. So here we saw two new terms here. So let's quickly list out what are the differences and what each one means. Okay. So prokaryotic. The word pro means primitive and karyon means nucleus. So prokaryotic cells, which means cells having primitive nucleus, and eukaryotic means the term you means true, so real. Okay, and karyon again means nucleus, so it has a true nucleus. So I told you, whenever there's a new term in biology, try to split the word and derive its meaning from the very word itself. Okay. So from the word prokaryotic cells, we saw the first point of difference is membrane bound nucleus is absent. So we said that there is a primitive nucleus. So the nucleus does not have a membrane around it. So the nucleus is free. It is naked. So the genetic material inside the nucleus is naked. Alright. So there is no nuclear membrane. Okay. And whereas eukaryotic cells have a membrane bound nucleus. The nucleus around has a nuclear membrane. Okay. Next, prokaryotic cells are smaller in size, whereas eukaryotic cells are larger. Prokaryotic cells have a single chromosome present, whereas eukaryotic cells, they can have more than one chromosome present in them. For example, human cells, how many chromosomes do we have? We have 23 pairs of 46 chromosomes, which means human cells are eukaryotic cells. And in prokaryotic cells, membrane bound organelles are absent. which means that the organelles like mitochondria, vacuole, plastids, and everything, they all have a membrane around them. So these all organelles are absent in them, whereas in eukaryotic cells, all these organelles are present. So this point is important to understand because this does not mean that organelles are absent in prokaryotic cells. Remember, ribosome is an organelle, but it is present in prokaryotic cells. Here I told you membrane bound organelles are absent. It does not mean organelles themselves are absent. Ribosome is a membraneless organelle, so it is present in prokaryotic cells. So organelles which do not have a membrane, they might be present in prokaryotic cells. Organelles with membrane, they are not present in prokaryotic cells. So ribosome, a special organelle, is present in both prokaryotic cells as well as eukaryotic cells. Now let's dive into prokaryotic cells and understand more. So as we saw already, they are smaller than eukaryotic cells, so they multiply more rapidly, more fast than eukaryotic cells. So some of the examples of prokaryotic cells are PPLO, which is Mycoplasma. PPLO stands for Pleuropneumonia-like organism. This is very special because it is the smallest organism found in nature and it's only 0.3 micrometers in length. And an important feature is that it does not have cell wall. So Mycoplasma is the only wall-less prokaryote. Very important. So all prokaryotes have a cell wall surrounding the cell membrane except in Mycoplasma. So this is an important fact that you should remember. Next, let's talk about bacteria. Bacteria is 3 to 5 micrometer in size. And we all know, I think you would have studied this in grade 9, the 4 basic shapes of bacteria, that is Bacillus, Coccus, Vibrio and Spirillum. Bacillus, rod shaped, Lactobacillus, the one present in your curd. It is rod shaped. Can you see this picture? This structure here is the spore. Important, remember the labeling. It is rod shaped. Next is coccus, spherical, it's round in shape. Next, vibrio, comma shaped. Next we have spirillum, spiral, it is spiral in shape. Look at this, it has a flagellum at its end. Flagellum is the motile structure which helps in movement. Now let's look at examples for eukaryotic cells. The largest isolated single cell is the egg of an ostrich, and this is again an important fact that you should remember from your NCERT. Okay. Next, let's talk about human RBCs, which are examples of eukaryotic cells. They are 7 micrometer in diameter, pretty large, and they are biconcave, not biconvex. Remember the terms, remember the spelling, it's important. It's biconcave and disc shaped. It's enucleated. What does this mean? Enucleated means the nucleus disappears, disintegrates at maturity. So this does not mean that nucleus is absent throughout its lifespan. No, that is not the meaning. That is anucleate. Here it is enucleated. Enucleated means at maturity the nucleus disappears. So talking about the lifespan of RBC, it's 120 days. And where does it get destroyed? It gets destroyed in spleen. Spleen, that organ, is commonly called as the graveyard of RBC. Okay, this is something extra which you'll learn little later. But remember, present in NCERT, so wherever there are relevant topics, I'm trying to connect all the points from your NEET syllabus. So whenever I say RBC, all these terms, biconcave, disc shaped, enucleated, all these should come to your mind. Okay. Next, nerve cell is again a eukaryotic cell. These are some of the longest cells. So we find that there are the shape and there are different shapes of cells. Why so? Because the shape of the cell corresponds to the function. So according to the different functions, the cell's shape varies structurally also. So the shape also matters here. How? Let's take an example, say RBC. I said it is biconcave and enucleate. Why? Because to maximize the space available to carry oxygen. That's why it's enucleated, so that there's more space available. Next, I talked about nerve cells, which I said it's long. This is how a nerve cell looks. So this is the cyton. Now I have the dendrites, rods of Ranvier, and all this. Okay. Now it is long so that impulse transmission occurs faster. And let's take another example, WBC. They are of irregular shape. They are like amoeboid shape. So why so? Because they perform the role of phagocytosis. They engulf foreign bodies, foreign particles, and they help in increasing our immunity. So the shape varies with the function of the cell. Okay. So let's talk about a prokaryotic cell more in detail. I told you prokaryote means primitive nucleus. So there is no well-defined nucleus. So such an not that much developed nucleus is known as nucleoid or nucleophore. We say it's not well defined because there is no nuclear membrane. So the nucleoplasm remains continuous with the cytoplasm of the cell. And we also saw that there are no membrane bound organelles, but ribosome, which is the only organelle present in prokaryotes, they are present. Okay, and they are membraneless. Okay. So since nucleus does not have a nuclear envelope, the genetic material is naked, and the genomic DNA is a single chromosome. We saw that there is only one chromosome, and it is circular DNA. Single chromosome, only one chromosome is present, and the DNA is circular. Important. Next, let's talk about plasmids. So these prokaryotic cells have an additional plasmid in them. What is a plasmid? Plasmid is extrachromosomal. Extrachromosomal means outside the genetic DNA, outside the main genetic material. A small circular DNA. So a plasmid is an extrachromosomal small circular DNA, and it confers unique phenotypic characters like resistance to antibiotics. Phenotype means external morphological features. So plasmid helps in providing certain unique features to the bacteria or any prokaryotic cell, and it helps to monitor bacterial transformation with foreign DNA. So about this you will learn in the chapter biotechnology, so you don't have to break your head too much on this. So the information about plasmids, they are important, remember. And I'm repeating this point again because it's extremely important that no organelles are found in prokaryotic cells except for ribosomes. Now prokaryotic cells also have a special feature in them, which is the mesosome. It is a characteristic feature of prokaryotes. It is a specialized, differentiated form. It forms the infolding of the cell membrane. So, the cell membrane, the plasma membrane, has certain infoldings into the cytoplasm. So, infoldings here means extensions of the plasma membrane into the cell, and these extensions can be in the form of vesicles, that are rounded bodies, tubules, which are like tubes, and lamellae, which are flattened sacs. Okay, these extensions can be of these forms, and these help in many functions like cell wall formation, DNA replication and distribution to the daughter cells, respiration, secretion, increase the surface area of the plasma membrane, and enzymatic content. So the extensions, the infoldings, help in cell wall formation, DNA replication and distribution to daughter cells, respiration, secretion, and they help in increase the surface area of the plasma membrane, and they help in increasing the enzymatic content. So how will you remember the functions of mesosome? So here I am with the trick. CD, sir. Okay, you can remember with this mnemonic. C, cell wall. D, DNA. Sir, S, secretion. I, increase surface area and enzymatic content. And R for respiration. So with this trick you can remember the functions of mesosome very easily. So they will give you these functions in the options maybe, and they will ask you which of these is an odd one out. So you can expect questions like that. For such cases, these tricks will be extremely handy for you to recollect. All right. Now we talk about structures called chromatophores present in prokaryotic cells. So these structures, so break the word and derive the meaning. So chroma means color and phores is something like extensions or sacs. Okay, so a body containing color which appear as extensions or sacs into the cytoplasm. That's it. So chromatophores are nothing but membranous extensions into the cytoplasm. These contain pigments, we said color or pigments, and these are present in some prokaryotes like cyanobacteria or blue-green algae. So cyanobacteria has an alternate term. Blue-green algae is mostly aquatic and it is photosynthetic, so which means it has chlorophyll in it and other pigments. That is all about chromatophores, a small topic. Next, there are bodies known as inclusion bodies. So, inclusion bodies' main function is to store reserve material. Important point here is that these inclusion bodies are not bound by any membrane system, so they lie free in the cytoplasm. So, what was the membraneless organelle we saw? Yes, it was ribosomes. Here, inclusion bodies, we are saying that they are not bound by any membrane system. So, two membraneless organelles or bodies are ribosomes and inclusion bodies. So presence of organelles in prokaryotes, yes, organelles can be present, but membrane bound organelles are absent. Membraneless organelles, yes, they can be present. So understanding the meaning is important. So examples of inclusion bodies are phosphate granules which store phosphate ions, cyanophycin granules, and glycogen granules which store glycogen or carbohydrates. Okay. Now prokaryotes have a unique cell envelope, and this cell envelope is modified into different means. So let's see what it is. So prokaryotes have a chemically complex cell envelope, and this cell envelope, I said it's chemically complex because it is composed of three layered tightly bound. Okay, all the three layers are tightly bound, and each layer performs a distinct function. However, they act together. Although each layer performs a distinct function, they have a unique function of their own. They act together as a single protective unit. So we call it together. We call all the three layers together as one envelope for the cell. So let's look what are the three layers. First layer is glycocalyx, which is the outermost layer. Next comes the cell wall, and the innermost layer is the plasma membrane. So GCP, glycocalyx, cell wall and plasma membrane. Glycocalyx is in contact with the outside environment, whereas the plasma membrane leads to the cytoplasm. So glycocalyx is something new here and we need to pay attention. So glycocalyx is that protective layer which is present in the outside, and it differs in composition and thickness among different bacteria. So I said composition and thickness. So it can be either a slime layer, which is a loose sheath, or it could be capsule, which is thick and tough. So this is how it differs in composition and thickness among different layer. So slime layer, loose. So look at the ending word, loose has S in it. Look at the pronunciation of the ending word, loose. So it is slime, SS. Thick how does it end? It ends with a CK. So look at the pronunciation, thick capsule. So you can remember it that way. Slime layer is loose. Slime itself, we know it's very loose, very you have played with slime, isn't it? Like that also you can remember. So slime layer is something loose, and capsule is thick and it is tough. The next layer of cell envelope is the cell wall. Cell wall determines the shape of the cell. It provides structural support and prevents bacterium from bursting or collapsing. So let's see how it helps in how it prevents the bacterium from bursting or collapsing. Say for example, we place a plant cell and an animal cell in an hypotonic solution. When we place it for some time, we find that the animal cell has burst. Okay, it bursts and collapses, whereas the plant cell remains turgid and stiff. This does not burst due to the presence of strong rigid cell wall. The reason why an animal cell bursts is that an animal cell has only a plasma membrane. It does not have a cell wall. So bacterium here is a prokaryotic cell. So even when you place a prokaryotic cell, it does not burst, okay, in a hypotonic solution. So because it has a rigid cell wall in it. All prokaryotes except mycoplasma has a cell wall. Remember, it's a fact. So what happens when you place mycoplasma in a hypotonic solution? You know it by now. Alright. Now let's talk about plasma membrane. Plasma membrane or cell membrane. It is selectively permeable in nature, which means it allows only certain or selective substances into the cell. Whereas cell wall, remember, it is fully permeable and there are no restrictions to it. It allows free movement in and out. Plasma membrane in prokaryotes is similar to that structurally to that of eukaryotes. This is very important MCQ. In MCQ point of view, this is an important point, that the plasma membrane of eukaryotes and prokaryotes are similar structurally. Okay. On the basis of differences in the cell envelopes and the manner in which they take up the staining procedure developed by Gram, a scientist, bacteria can be classified into two groups, gram positive and gram negative bacteria. So Gram, he was a scientist, he developed a gram staining procedure. So gram positive, just by the name you can say that they take up the stain, and gram negative are the bacteria which do not take up the stain. Simple. Gram positive bacteria, their cell wall is composed of thick layers of peptidoglycan. So this peptidoglycan is responsible to take up the stain. And for gram negative bacteria, the cell wall has thin layer of peptidoglycan. So you might be wondering, what is peptidoglycan? So please note here, the cell wall of plants is made up of cellulose, whereas cell wall of fungi is made up of chitin, and cell wall of bacteria is made up of murin or peptidoglycan. Remember. So what is the cell wall of bacteria made up of? Murin or peptidoglycan. So it is thick in gram positive and thin in gram negative. So hence they do not retain the purple color stain. The gram negative bacteria do not retain the purple color stain in the gram staining procedure. Okay. Now bacteria has certain surface structures like flagella, pili, fimbriae. Flagella plays an important role in motility, whereas pili and fimbriae do not play any role in motility. Here you need to remember this important. Remember, flagella is present both in prokaryotes as well as eukaryotes. Prokaryotic flagella arises from the cell wall, whereas eukaryotes it arises from the plasma membrane. Prokaryotic flagella is simple. it is composed of a flagellin protein, whereas eukaryotic flagella is a little complex and it arises from the basal body of centriole. So about this, we'll learn more in upcoming classes. Okay. Let's talk about bacterial flagella, the flagella present in prokaryotes. I told you it helps in motility. It is thin filamentous extensions from their cell wall, not plasma membrane. Remember, important sentence from the NCERT. And it is composed of three parts: the filament, hook, and basal body. Filament is the longest portion, and it extends from the cell to the outside surface. So now I'll draw the structure so that you understand. This part is the filament, which is the longest part, and this part forms the hook. And we have three rings, LPSM rings. These together comprise the basal body. So here you see the L ring, it is the cell wall, and the last ring, that M ring, here comes the cell membrane. This is how a prokaryotic, a bacterial flagellum looks like. So remember that bacterial flagellum arises from the cell wall. It's an NCERT line, remember. Now let's move on to the next surface structure, which is pili. Pili is an elongated tubular structure made up of a protein known as pillin. Like flagellin, here it is pillin. That's it. So pili is elongated. It is long and it is tubular. Okay. Next surface structure is fimbriae. Fimbriae is small. Pili was long. Fimbriae is small and it is bristle-like fibers, an important phrase which gets repeated in the question papers. And these fimbriae help in attachment of the bacteria to rocks in streams and to the tissue of the host organism. So these were all about the surface structure of bacteria. Next, we are going to talk about the special organelle which is present in both prokaryote as well as eukaryote, which is ribosomes. Ribosomes, I said, they are membraneless, and these were first observed by George Pullett, so they are also known as Pullett particles. Ribosomes are composed of RNA and protein. Important. Ribosomes are site of protein synthesis, which means in ribosome there is synthesis of protein. So ribosomes are most commonly referred to as the protein factory of the cell. Control room of the cell is nucleus. Protein factory of the cell is ribosome. Recollect the alternate names. Okay. So several ribosomes attach to a single mRNA to form a chain known as polyribosome or polysome. So this is a ribosome which has two units, a smaller and a larger subunit, and these ribosomes get attached to a single mRNA, and this together is known as a polysome. So what is a polysome? Ribosome plus mRNA. Many ribosomes plus a single mRNA comprises a polysome. So they might twist and turn. They'll say one ribosome and many mRNA, things like that. So just remember this diagram. It will help you not get confused with the lines given in your question paper. So visualize and learn. Okay. These ribosome of a polysome translate the mRNA into proteins. So we will learn the process of translation little later. Okay. So this is how, but by becoming the polysome, they are able to synthesize protein. That much if you understand, it's enough. Why is this? Why are the ribosomes going and attaching to the singular mRNA? For protein translation. That much if you understand, it is enough. Okay. Okay. And the size of ribosomes are just 15 nanometers to 20 nanometers. Okay. Now, I told you that ribosomes are present in both prokaryotes and eukaryotes, but they are not the same. Let's look at their differences. Prokaryotic ribosomes are associated with the plasma membrane of the cell, whereas eukaryotic ribosomes are found in cytoplasm. They are found within the organelles like chloroplast and mitochondria, and they are found on the organelle endoplasmic reticulum ER. Prokaryotic ribosomes are smaller, they are 70S in type, and eukaryotic ribosomes are larger, they are of 80S type. The 70S ribosomes, they are in turn composed of two subunits, 50S and 30S. 50S being the larger and 30S being the smaller subunit, obvious. And next, eukaryotic ribosome, the 80S ribosome is in turn composed of two subunits, which is 60S ribosomes and 40S ribosome. So you might be wondering what this S stands for. S stands for sedimentation coefficient. The S represents Swedberg's unit. So S is a unit known as Swedberg's unit. And what does it stand for? It stands for the sedimentation coefficient. This is an indirect measure, not direct measure. Remember, these words are also important. They are indirect measure of density and size. Okay. Now let's look at a prokaryotic cell, a bacterial cell, and try to recollect the components of the cell. So just by looking at the cell, we have the outermost glycocalyx, the capsule. Capsule is thick and tough. Okay, next comes the cell wall, next is the plasma membranes, and these three layers together comprise of the single protective unit known as the cell envelope. Next is the cytoplasm, semi-fluid gel-like matrix. Next, these small rounded particles are the ribosomes, which are of 70S or 80S? 70S. Next, we have plasmin, which is extra chromosomal. Can you see it's outside the genetic material? It's something other than genetic material. Okay, here next, the surface structure we can see is pili. This does not help in motility. These long one you see, this is the bacterial flagella, that helps in motility. The main genetic material you see, this is the nucleus of the cell. This is not enveloped by a nuclear membrane, so it is a prokaryotic cell, which means the nucleus is primitive. Okay, not well defined, so we call it a nucleoid or nucleophore. And this nucleophore has circular DNA in it. Can you see circular DNA? This is how a circular DNA looks like. The plasmid is also a circular double-stranded DNA, but it is extra chromosomal. It's outside the main genetic material. All right. Now we have something known as gas vacuoles, which are found in blue-green and purple and green photosynthetic bacteria. Gas vacuoles or pseudo-vacuoles we call it, because they are membraneless. Why gas? Because gas provides buoyancy for aquatic bacteria. So this was all about prokaryotic cell. Now is the time to talk about eukaryotic cells. So from this picture, what do you observe? A plasma membrane and outer covering? Yes. The central nucleus with nuclear membrane? Yes. And these are all the different cell organelles, membrane bound cell organelles. So eukaryotic cells occur in protists, in bodies of plants, animals and fungi. We know Whittaker's 5-kingdom classification, isn't it? There is Monera first, then Protista, fungi, plantae and animalia. All these are eukaryotes except Monera, which is a prokaryote. Okay. Now let's look at the features of eukaryotic cell. Eukaryotic cells have extensive compartmentalization of the cytoplasm, which means the cytoplasm is divided into different compartments by means of membrane bound organelles. The membranes of the organelles divides these or separates it from the cytoplasm. So which means the matrix present inside the organelle is not continuous with that of the cytoplasm of the cell. And there is an organized nucleus with a nuclear envelope. Yes, we already saw. And there are complex locomotory structures and cytoskeletal structures. So, unlike prokaryotic flagella, which is made up of flagellin protein, the flagella present in eukaryotes, I told you it is complex. Cilia, flagella, they are all complex structurally, and there are cytoskeletal elements which we will learn later, and they are unique features of eukaryotes. And genetic material is organized into chromosome. Genetic material is not naked because it is covered with a nuclear envelope, and they get organized into condensed form known as chromosome. So now I would like to ask you a question. Are all eukaryotic cells identical? True or false? Tell me. All eukaryotic cells are identical. True or false? It's the NCERT line. Please comment your answers. I'm not going to answer this question. And very, very obvious question. Okay. So now, just as an answer to the question I asked you, let's compare plant cells and animal cells. By now you would have understood. We are going to look at the differences in plant cell and animal cells. Plant cell wall is made up of cellulose, and in animal cells there is no cell wall, there is only plasma membrane. And plant cells have one large vacuole which occupies 90% of the cell volume, whereas in animal cells there are several small vacuoles. There is not a big vacuole present. Plant cells have plastids present in them, whereas animal cells there are no plastids. Plant cells do not have centrosomes or centrioles, whereas animal cells they have 2 units of centrioles. In plant cells, nucleus occupies the periphery position because the vacuole occupies 90% of the volume of the cell. It is central in position, so automatically the nucleus is pushed to the periphery, whereas in animal cell it is the nucleus which is occupying the center of the cell. In plant cell, cytokinesis, that is division of the cytoplasm, occurs by cell plate formation from middle, that is from the center, to periphery. So it's away from the center, so it is centrifugal in nature, we say. So in animal cells, cytokinesis, that is division of cytoplasm, takes place by cleavage furrow formation from periphery towards the center. So which means center seeking, which means it is centripetal. So remember, important difference: cytokinesis in plant cells occurs by cell plate formation, that is which arises from Golgi apparatus or the phragmoplast, we say, that is centrifugal in nature, away from the center, whereas in animal cells it occurs by cleavage furrow formation, centripetal in nature. Okay, now this is a diagram of plant cell, and let's try to spot on the different organelles present. Here, just by looking, we find a large central vacuole occupying 90% of the cell. It's an important data, please remember. Next we have cell wall. Next we have the green chloroplast containing chlorophyll, which is responsible for photosynthesis. Next we have the semifluid matrix cytoplasm. Next mitochondria, which we call the powerhouse of the cell. And next we have Golgi apparatus. Here you can see, yes, this is the Golgi apparatus. And few rounded bodies near the Golgi apparatus, they are known as Golgi vesicles. They are rounded spherical bodies. And here you see they are the ribosomes. And we find that the nucleus is pushed to the periphery as discussed earlier. And near to the nucleus we have endoplasmic reticulum, rough endoplasmic reticulum bearing ribosomes on it, and smooth endoplasm reticulum, they do not have ribosomes. So endoplasmic reticulum, remember, it's an organelle which is present just near the nucleus. Next we have cell membrane, which is inner to the cell membrane, which is inner to the cell wall. Okay. Next here we find cytoskeletal elements composed of 3 things: microfilaments, microtubules, intermediate fibers, filaments. More about this we'll learn later. Okay. Now this cell here is the animal cell. And quickly let's discuss the organelles we can spot here. First of all, we have the cell membrane, the outermost covering. Next, we have the central nucleus, and inside the nucleus, we have a nucleolus, which is the rounded body present inside the nucleus, and this nucleus is covered by a nuclear membrane. Now, we have a structure here which was unique, which was not present in plant cells, and that is the centrosome. Here is the smooth endoplasmic reticulum near the nucleus, and here there is rough endoplasmic reticulum bearing the ribosomes. Then we see the mitochondria. Yes, here we see several small vacuoles which are pushed to the periphery by the centrally occupying nucleus. Here we see cytoplasm and ribosomes, and here we also see a structure, an organelle known as lysosome. So in plant cells, the lysosome is known as spherosomes, which are derived from smooth endoplasmic reticulum. Spherosome is a microbody. So there is something known as a dictyosome. So dictyosomes are plant Golgi bodies. We call it by a special name because the Golgi bodies are present as unconnected units, unlike in animal cells. So two terms we discussed: plant Golgi bodies and plant lysosome, which is dictyosome and spherosome respectively. Spherosome are a part of microbodies. Microbodies include peroxisomes, spherosomes, and glyoxysomes. These three comprise the microbodies. Okay, so we were talking about cell membrane, which is common to both plant cells and animal cells. We also saw that cell membrane of prokaryotes and eukaryotes are structurally similar. Now let's look at how the cell membrane was studied earlier initially. How the detailed structure was revealed. The detailed structure of cell membrane was revealed only after the electron microscope came into the picture in 1950s. The cell membrane, when studied chemically, especially when we studied the cell membrane of RBCs, we were able to deduce the structure of the plasma membrane. And from the chemical studies, we came to know that cell membrane is composed of many lipids and proteins and also cholesterol. So what does the plasma membrane or cell membrane composed of? Lipids, proteins and cholesterol. As of now, only this much we've seen. Okay. And these lipids are phospholipids. The majority of the lipids are phospholipids, which are arranged in a bilayer, that is two layers, with the polar hydrophilic. Hydrophilic means water loving head towards the outer side. Polar hydrophilic, so loving head, okay, towards the outer side, and non-polar hydrophobic. Hydrophobic means water hating. Phobic. Philic means affinity towards water. Phobic means fear towards water. That tail lies towards the inner part. This is how, let me draw. And this way it looks like a bilayer. Okay. Head on the outer side, tail on the inner side. So that the tail, which is made up of saturated hydrocarbons, is protected from the aqueous environment. Remember, saturated hydrocarbons, not unsaturated. They'll mix up and they'll confuse you. Okay. Then, after chemical studies, biochemical investigations were done, and this revealed that cell membrane is also composed of protein and carbohydrate. So what does the cell membrane composed? We saw lipids, proteins, cholesterol and carbohydrates. Okay. Lipids, protein, cholesterol were found from chemical methods, and proteins and carbohydrates were found from biochemical methods. This is what these methods revealed. Okay. And now you should understand that the ratio of protein and lipid is different in different cells. In RBC, erythrocyte, erythro means red, cyte means cell, red cells, red blood cells. Protein is 52% and lipids are 40%. So this is an important factual information that you should remember. And depending on how the proteins can be extracted from the membrane, the membrane proteins can be classified into two types: integral and peripheral proteins. As the name suggests, integral means lies the interior, whether it is partially or totally buried or embedded in the membrane. Important, it can be partially or totally buried. Whereas peripheral, it lies totally towards the exterior, it lies on the surface of the membrane. It was Singer and Nicholson who proposed the fluid mosaic model of the plasma membrane in 1972, which is a very popular model, and from that we can get a clear picture of the cell membrane. And they proposed the fluid mosaic model. They said the quasi means semi-fluid nature of lipids enables lateral sideways movement of protein. Important, very very important sentence. Quasi-fluid nature of proteins enable lateral movement of lipids. Is this sentence right? Nope. This is incorrect. LL, lateral movement of lipids. This can never happen. So LL does not come together. So remember, you can remember it this way. So be careful. They'll mix up the words and they will confuse you. So remember, this is an important sentence. Quasi-fluid nature of lipids enables lateral sideways movement of protein within the overall bilayer. And this enables the fluidity of the cell membrane. And this fluid nature in turn enables the cell to perform various functions like cell growth, formation of intercellular junctions, secretions, endocytosis, and cell division. This fluidity of cells is also responsible for transport within the cell. We already saw that the cell membrane is selectively permeable, and this enables different kinds of transport, allows certain substances into the cell either by passive transport. So by passive transport we mean that there is no requirement of energy for that transport, and this passive transport is just by simple diffusion along the concentration gradient. That is, molecules move from higher concentration to lower concentration, always to high to low. So this is what we mean by along the concentration gradient. There is a gradient from high to low, and this is just by simple diffusion. For example, movement of neutral solutes and water by osmosis. So osmosis is a special type of diffusion. Apart from passive transport, we have facilitated transport. As the name suggests, facilitated by a carrier protein. So the polar molecules, some of the polar molecules cannot pass through the non-polar lipid bilayer. We know that the lipid bilayer composed of the head and the tail. So what is inside is the tail, which is non-polar. So polar molecules cannot easily enter the lipid bilayer. So for this they require the help of a carrier protein. So with this carrier protein, it facilitates the transport of that polar molecule across the membrane. Next type of transport is active transport. Active means energy dependent. And why do we need energy? Because it is against concentration gradient. Uphill we say. So remember, there are two terms here, against and along. Against concentration gradient, that is from lower to higher concentration, it is for active transport. And along concentration gradient, higher to lower, is passive transport. So don't get confused. So uphill. So remember, you're climbing a hill up. You require more energy to climb a hill up. Rather, when you climb down, it's a sloping surface, so you can just flow in through, you can just go along the surface, so it's much easier, so you do not require much energy to climb down. Example of active transport is sodium potassium pump. So this is a table which is given in transport in plant chapter. So I thought of adding it here itself because it will become easier, because you're learning the concept here. So it will be easier to learn this table as well. So let's talk about the property and then about the different types of transports. Special membrane proteins are not required for simple diffusion or passive transport. There is no such requirement of special proteins. Whereas facilitated protein and active transport, yes, there is a requirement of carrier protein. Simple diffusion, no, not at all. It's not at all selective. Whereas facilitated transport and active transport, yes, it is very much selective in nature. It is selective on the different membrane proteins, the different substances, the different molecules it transports across it. Does this transport saturates? No, it does not saturate in simple diffusion. What does it mean by saturation? Transport saturation? The ability of the carrier protein to carry that molecule across the membrane. It does not have a membrane protein itself, so how does the transport get saturated? No. So simple diffusion, no. But yes, for facilitated transport and active transport. Whether it is an uphill transport, no, for simple diffusion. It is downhill, it is along the concentration gradient. Facilitated transport, is it uphill or downhill? It is downhill. It is just same as passive transport, but just that it requires a carrier protein so that polar molecules can pass through the non-polar lipid bilayer. Whereas for an active transport, yes, it name itself suggests that active, so it is uphill. Same requires ATP energy. No, simple diffusion does not require. Facilitated transport does not require energy. Whereas active transport is uphill, so it requires energy for that transport. Remember, facilitated transport is along the concentration gradient without requirement of energy. So here I want to show you the actual how the actual structure of the lipid bilayer of the plasma membrane looks. So here we can see two layers, so the name bilayer, and composed of phospholipids, so the name phospholipid bilayer. So first thing we can observe is the peripheral protein, which is on the outside, clearly present on the outside, so it is peripheral. And these small structures are nothing but sugar or carbohydrates. So we can say plasma membrane is composed of carbohydrates also, and this was revealed by biochemical methods. Okay, investigations. Okay. So here we see hydrophilic head and hydrophobic tail. The hydrophilic tail is protected from the aqueous environment. Yes, and here we see two proteins here, which are the integral proteins. One which is totally embedded inside, which is embedded inside the lipid bilayer. Yes, and this you see it is partially embedded. Some part is outside also. So some part is inside, some part is outside. So partially outside, partially inside. So we say this is partially embedded. And here you see there is cholesterol in it. So this was the one which I stressed on. And this is an important line you need to remember. Quasi-fluid nature of lipids help in lateral movement of proteins. Yes, LL does not come. So remember that way. And what are the functions enabled by the fluid nature of plasma membrane? Growth and cell division. Growth of cell and cell division. Okay. Cell growth and division. Second, secretion. Next is endocytosis, transport, or next fourth one is formation of intercellular junctions. That's it. Just a quick recap. We already talked about cell wall when we discussed about prokaryotic cells, and here same thing again. Let's recap. It is non-living. That's the one point we missed out. So remember, cell wall is non-living. It's a rigid structure found as an outer covering in plants, animals, and all prokaryotes except mycoplasma. It gives shape and protects the cell from mechanical damage and infection. It helps cell to cell interaction and provides barrier to undesirable macromolecules. Remember it as SIP, shape, structural support, interaction, cell to cell interaction, and P for protection. An algal cell is made up of cellulose, galactans, mannans, and minerals like calcium carbonate. Important, remember. And plant cell wall is made up of cellulose, hemicellulose, pectin, and proteins. Cellulose, galactans, mannans, and calcium carbonate. It's for algal cell wall. Plant cell wall, it is cellulose, hemicellulose, pectin, and protein. Okay. So now there are two kinds of cell wall. One is the primary cell wall and the other is the secondary cell wall. Primary cell wall is the cell wall which is present in young plant cells, and this primary cell wall is capable of growth. So as the cell mature, this primary cell wall gradually diminishes, gradually disappears, and the secondary cell wall now forms on the inner side, that is inner to the primary cell wall when the cell attains maturity. So first you have this primary cell wall and then inner to the primary cell wall. This first primary cell wall, this is found when the plant is young. Okay, then as the plant matures, what develops is the secondary cell wall inner to the primary cell wall, and this structure you see is the plasma membrane or cell membrane. So the order from outside to inside is important. And here is the cytoplasm, vacuole, and other organelles like mitochondria, nucleus, dictyosomes, ribosomes, lysosomes, and so on. Next structure is the middle lamella. It is a cementing material made up of calcium pectate. The main function of middle lamella is to hold the neighboring cells together. So it connects the cell wall of two adjacent cells and it acts like a cementing material holding the adjacent cells together. Okay. So this cell wall and the middle lamina, they may be traversed or cut across. We say traversed meaning travel across or crossed by plasmodesmata which connect the cytoplasm of neighboring cell. So remember, middle lamella connects the cell wall of neighboring cells and plasmodesmata connects the cytoplasm of neighboring cells. So this differentiation is important. And it is maybe. So plasmodesmata may be present or absent in some places of the cell wall and middle lamella. Okay. Now we move on to talk about organelles. Firstly, we deal with endomembrane system. Endomembrane system includes four organelles: endoplasmic reticulum, Golgi complex, lysosomes, and vacuoles, because these organelles act as a single unit. Their functions are coordinated. Okay, their functions complement one another. So let's look at the first organelle, which is endoplasmic reticulum. So endoplasmic reticulum, the name reticulum means network. Okay, this endoplasmic reticulum divides the intracellular space, the space inside the cell, into two distinct compartments. One is the luminal, the other is the extraluminal. Luminal is inside the ER. So endoplasmic reticulum divides the cell into two compartments. One is inside endoplasmic reticulum and the other part is outside endoplasmic reticulum, that is the cytoplasmic components. Okay, so it is luminal and extraluminal. Endoplasmic reticulum may be of two types. one rough endoplasmic reticulum. Next is the smooth endoplasmic reticulum. We have already seen rough. The surface is rough because it has ribosomes present on the surface when viewed under the microscope. Whereas the surface is smooth because of the absence of ribosomes. Hence, the surface appears smooth. Rough endoplasmic reticulum, we see the endoplasmic reticulum itself. I told you it is near to the nucleus. It is present near the nucleus. Okay. So, talking about rough endoplasmic reticulum, since it is associated with ribosomes, they are found in cells which are actively involved in protein synthesis and secretion. And these rough endoplasmic reticulum, they are extensive and continuous with the nuclear membrane, the outer membrane of the nucleus. And talking about smooth endoplasmic reticulum, the main function is synthesis of lipid. For rough endoplasmic reticulum, main function was protein synthesis. For smooth endoplasmic reticulum, main function was lipid synthesis. And in animal cells, lipid-like steroidal hormones are synthesized in smooth endoplasmic reticulum. So, lipid synthesis and lipid-like steroidal hormones are site of synthesis in smooth endoplasmic reticulum. Next component of the endomembrane system is the Golgi apparatus. This was first observed by Camillo Golgi as a densely stained reticular structure near the nucleus. So remember, Golgi apparatus are densely stained and they are found near the nucleus. Golgi apparatus has flat disc-shaped sacs parallel to each other, and this structure is known as the cisternae. This is just 0.5 micrometer to 1 micrometer in diameter, and this cisternae has two faces. The convex or cis or forming face, the alternate names are important, and the concave, trans or maturing face. So both these phases are entirely different but interconnected. An important sentence: these phases of cisternae, they are different in functions they perform, but they are interconnected. Let's look at the functions of Golgi apparatus. Main function is packaging materials in the form of vesicles from ER. So can you see that their functions, the functions of ER and Golgi apparatus are coordinated? So the vesicles, okay, from ER, they are packaged by Golgi apparatus. The vesicles from ER fuse with the cis face of Golgi apparatus. So let me draw here so for you to understand better. They fuse with the cis face of the Golgi apparatus. They move towards the maturing phase, and finally they are released as packaged materials. So it is because the Golgi apparatus remains in close association with the endoplasmic reticulum. Both of these functions are coordinated, so we say that they are a part of endomembrane system. Next function of Golgi apparatus is that it is the site for formation of glycoproteins. So this process of formation of glycoproteins is glycosylation, and the formation of glycolipids is glycosidation. Silation, I told you LL does not come together. So silation is for proteins and not lipids. Remember that way. Glycosidation for glycolipids, glycosylation for glycoproteins. Okay. Now we move on to the next organelle under endomembrane system, which is the lysosome. Lysosome is formed by the packaging in the Golgi apparatus. So lysosome is one of the packaged product of Golgi apparatus. So this is an important function. So Now it is clear that while lysosome is also a part of endomembrane system, isn't it? And this lysosome is rich in hydrolytic enzymes which are active in acidic pH. So hydrolytic enzymes include hydrolases like lipases, proteases, carbohydrates which digest carbohydrates, proteins, lipids and other nutrients like nucleic acids and so on. So basically they digest the materials, hence they are known as suicidal bags of the cell. They are also capable of auto digestion. They can digest themselves because of the hydrolytic acidic enzymes present. The last organelle of the endomembrane system is the vacuole. Vacuole contains sap, water, excretory products and other materials not useful for the cell. So vacuoles are just storehouse of the cell. They store waste material of the cell, and this is bound by a single membrane which has a special name called tonoplast. And as I told you earlier, it occupies 90% of the space in the plant cell. So tonoplast, I told you, it serves a special function. It facilitates transport. Tonoplast is just the cell membrane. Okay, it's just the membrane of vacuola. So it facilitates just like cell membrane, so it facilitates transport. But what kind of transport? It is active transport. Why? Because the materials are transported against the concentration gradient into the vacuole, which means that vacuole has higher concentration than cytoplasm. That is, the sap inside the vacuole is hypertonic to that in the cytoplasm. Understood? That is why transport of molecules, ions, takes place against the concentration gradient. So this is an important point and this is an example of active transport. There are different types of vacuole. In amoeba, there is contractile vacuole which helps in osmoregulation and excretion. Osmoregulation is nothing but maintaining balance like acid base balance, salt water balance. All the balance, the regulation is maintained by contractile vacuole. Similarly in protists, okay, there is food vacuole which is formed by engulfing the food particles. So when a food particle is there, the protista engulfs the food, tries to engulf the food particle, and in that process a food vacuole is formed. Okay, next, as we saw earlier, there is gas vacuoles or pseudo vacuoles. They are membraneless and they provide buoyancy to eukaryotes. Alright, so this was all for today's class. This lesson is important for forming the basis of the entire botany. So more about the other organelles we shall see in our next class. Until then, stay tuned. Thank you so much for watching.